now it's my big pleasure to introduce Margherita. Margherita is, she has her lab in Switzerland, but she's originally from Italy, where she started studying something that is not really related, but related to what she does now. She's a superstar in the field of human placenta development and how environment affects this in this model that we were hearing about organoids. She got like really prestigious awards in Europe. She was one of the Marie grants that are really, really tough to get in Europe. She's also part of an awardee of a L'Oreal Women in Science. And she's traveling the world, so she will present her talk and then catch a flight to go to another conference in Paris. So, Margherita.
Thank you, Miguel, for the really kind introduction. And good afternoon, everyone. It's truly an honor to be here. When we think about the strides science has made in our advancements in understanding of the human body, it's really incredible. But as we've heard and as we know, when it comes to female physiology, there's still so much left to uncover. And it's been really inspiring hearing the talks here since yesterday about how people are trying to make a difference in this area. And it's wonderful also to be at a conference where I don't need to convince people that it's an important area to do research. So I'd like us to imagine a future where we have really uncovered the intricacies of the female physiology. A future where we have really precise tools, where we can diagnose conditions early and find really the effective medical treatments to treat these conditions. So 10 years ago, when I was beginning to set up the culture systems to study the human endometrium and the placenta, with the systems that I'll be sharing with you today, I didn't think that we'd be here where we are now. So it's thrilling to see, as I said, to see how far we've come as a community, and to witness really the insights and the breakthroughs that we're all achieving in our individual fields. And I also, I've been really deeply inspired. So again, I'm really grateful, and I'd like to thank the organizers, particularly Yoel, for having me here. And I'm excited to share with you our latest discoveries and give you a basic scientist perspective on how we are trying to understand and how, placental biology and uterine biology, and how we can use organoids to do this. So let's start. So the central question that we are addressing in my lab is really trying to understand how the human placenta develops and how this process is influenced by its interactions with the maternal uterine microenvironment. So in the previous talks, we've heard about germ cell development, early embryonic development, and I'm going to be telling you about a process that's happening several weeks later, in the first few weeks after implantation, where this really important process of these maternal and fetal cells intermingling with one another and forming this sort of, arriving to this compromise and forming this functional unit over this short and defined period of time that is pregnancy. And as a fundamental researcher, we're really interested in the building blocks of this process, understanding the molecular and cellular mechanisms and how this all works. So the placenta is a really fascinating organ. It's so diverse across the mammalian species. And human placentation is rather unique because it is a highly invasive process. So in a normal presentation, as you can see here, we see the villi of the placenta embedding into the uterine wall, and I've just simplified and represented here by dots these invasive specialized placental cells that migrate through the uterus through the first layer and into the second layer, that is the myometrium, where it stops. And these invasive placental cells have the really important task of transforming the maternal environment, particularly into tapping into the maternal blood supply to ensure enough exchange and resources for the fetus, the placenta, and therefore the fetus to grow. And this process of invasion, this balanced process, is really important for proper placentation and successful pregnancy, because it is in the two extremes of invasion that we see the major pregnancy disorders. So on one side, in the placenta-accreta spectrum, we have the placenta basically invading through the uterine tissue almost like a tumor, destroying it, and it can also invade surrounding tissues. And on the other, a rather disordered and inefficient invasion of the trophoblast is characteristic of miscarriage, preeclampsia, growth restriction, and so on. And around 10 to 15 pregnancies, first pregnancies, are affected by these disorders. So if we think about what this means for a moment, it means that we all know someone that has experienced these conditions if we haven't experienced it ourselves. So why is it that we understand so little about this? And a conversation that I had with an obstetrician really stuck in my mind, where she told me that what's really hard is when a patient experiencing these distressing conditions then asks her, but why does this happen? And she doesn't have an answer. So that is our goal as fundamental researchers. We really want to fill this knowledge gap of understanding how this all works, how these two maternal and fetal cell types communicate and achieve this incredible feat that happens during pregnancy. And as I said, this has been a really difficult area of research. We are looking at a really highly complex and dynamic process that is happening in an early developmental time window. And as I said, not all these human-specific features are fully recapitulated in the common animal models and in vitro tools that we had that were used until till now. But what has really is changing the way we can study this really inaccessible process are the use of organoids. And as we've heard already this morning, organoids can be derived from stem cells, iPS cells, or human ES cells, but the organoids that I will be telling you about today are derived directly from tissues. And what is really powerful about this system, which has been established by the Hans Cleaver's lab back in 2009 for the intestine, and now we have organoids from all the major organs, is that by culturing the cells in the specific conditions that the tissue needs, they self-organize into 3D structures that resemble basically miniature versions of the tissue. And in this way, they really recapitulate the important functional properties that normally 2D systems cannot really recapitulate. And on the other side, it is a reductionist system, so it has the advantages, as I will show you, of not having to work with in vivo models. But of course, this doesn't mean that either of the tools are not useful and we just have to work with organoids. It just depends on the kind of question you're answering. And for us, it has been really, really important for our research. And there are many researchers out there using different types of organoids from different organs, and you can really use these systems to study, for example, viral and bacterial infections, diseases such as cancers. You can study cell-to-cell interactions. You can also freeze them and biobank them, so you can recover organoids that you've established to do studies when you want, and you can expand them to do high-throughput studies such as drug screening. So it's really transforming the way we're studying human tissue in health and disease. So for my talk, I'd like to tell you about the systems, the organoid systems of the endometrium, which is the lining of the uterus, and of the placenta, which we call trophoblast organoids, and give you some snapshots of how we are using this to address our central question. Then at the end, I will discuss a little bit about where I see are the future directions and the challenges that we still face in our field. So first, the maternal side of the story. So I would like to give you an example of how we are using organoids to study how the epithelial lining of the endometrium cycles at every menstrual cycle and how it repairs and how it prepares for pregnancy. So just as a reminder, the endometrium is the inner lining of the uterus, and it is a highly complex and dynamic tissue that undergoes shedding, growth, and differentiation and preparation for pregnancy, and this happens around 400 times in a woman's life, and the shedding occurs without any, and the repair occurs without any scarring, so it's an incredible tissue with regenerative capacities. And it contains many different cell types. It has epithelial cells, stromal cells, immune cells, but my focus of my talk will be these glandular structures, the epithelial cells. And the endometrium has the really fundamental role in pregnancy because it is the site of implantation. And once implantation occurs, this is where the placenta is embedded. You can see it here. And the secretions from the glands are thought to produce important signals for sustaining and the growth of the placenta. And we can capture the epithelial population of the endometrium using the organoid technology. And this method consists in basically taking biopsies from the endometrium. We basically process the tissue to release fragments of the cells that we're interested in. And then these are embedded into this gel-like droplet that we call matrigel, which essentially provides that 3D tissue-like environment for the cells to grow in. And using specific media conditions that allow the cells to survive and grow, we can expand the epithelial population of the tissue through these structures called organoids. They're really beautiful. They look like bubbles. They're made of many different cells. And essentially, we can keep them growing in a dish and get a lot of material to do experiments. And this is just to show you that these cultures are highly proliferative. So you can see here by the yellow dots, each one is a cell, nucleus of a cell, and it's marking the cells that are undergoing proliferation, so they're growing. And this derivation process is really highly efficient. We've now derived more than 80 lines from different samples. And what is also really powerful about this system is that not only can we just expand these stem cells to do experiments, but then they are able to respond to cues as they would do in vivo. And the menstrual cycle is dominated by two ovarian hormones, estrogen and progesterone. So estrogen is important for the regeneration and growth of the endometrium upon menstruation, and progesterone is responsible for basically preparing the endometrial lining for pregnancy. So we can get our organoids and mimic these hormonal signals and give them these hormonal signals, and they will respond. And essentially, they go transition from proliferative stem cells into generating differentiated cells. So here we see in red these cilia structures, which are these hair-like structures on cells, which is important for wafting secretions. And these cells are characteristic of the luminal surface where implantation occurs. We see them here in vivo. And here we see these secretory cells, which make up the bulk of the glands here, which produce these important secretions, again, for implantation and for placental growth. And you can see the corresponding cells here in vivo. And so it allows us to study the emergence of these specialized cells, that it's important for the function of the endometrium. But then we are also able to introduce additional steps into this protocol in order to mimic the menstrual cycle. So during the menstrual, if there is no pregnancy, progesterone and estrogen levels drop, and we can mimic this hormonal withdrawal in culture, and there is a breakdown of the tissue in vivo, and we can mimic this in vitro by basically mechanically breaking down the organoids into fragments, and you can see them here. And then we can start the cycle again by reintroducing the hormones and repeat this cycle over and over again so that we can mimic at the epithelial level what is happening in vivo. And because this protocol has really discrete steps, it allows us to ask questions about specific questions about how this epithelium responds to hormones, for example. And we can also ask about how it undergoes when it undergoes breakdown, how does it repair? This really important process that essentially marks the rest of the cycle, because if the endometrial lining does not repair properly, then it's not going to manage and to undergo the rest of the cycle, and there are many disorders of the endometrium linked to this. And so I just want to give you an example of how, for example, we could try to address what's happening, what are the drivers of this process of the repair in vitro. So again, here are the organoids that have seen the hormones. They are producing a lot of secretions. This is why they have this kind of black core. And essentially, so we then withdraw the hormones and break them up into little fragments, allow them to reform the organoids and undergo this repair process. And we've characterized this by RNA sequencing to identify some of the genes that may be involved in this process. And when we look at the genes that are upregulated in this organoids undergoing repair, we see a lot of processes that we know and expect to see in menstruation, during menstruation, which is, for example, the epithelial migration, regulation of fibrinolysis, and inflammatory responses, which is important for the influx of immune cells, which are highly involved in this repair process during menstruation. And so, as an example, what we can also do is then from this list of genes that we find are highly expressed in the organoids undergoing repair, we can take, and here is an example, we took Wnt7a, and Wnt7a is known also from mouse models to be important for uterine patterning, gland formation. So we were really interested in understanding what this gene may be doing in this repair process. And we can see here, this is a cross-section of an organoid after hormonal withdrawal, and this is after repair. Each dot, green dot, represents a transcript of that gene. And this is just the quantification to show you before and after repair that there's an increase in expression. And then you can use the organoids, genetically manipulate them. We can delete Wnt7a. And from the organoids, and you can see and ask what is the effect? Does it really, do we see something about an effect on the repair? And indeed, the wild-type organoids are able to grow again and repair normally, but the knockout really is a really drastic phenotype where the organoids cannot reform and grow. So in this way, we can use this rather reductionist system to start really looking at specific genes and drivers of key processes occurring during the menstrual cycle. So for this part, I showed you that the organoids endometrial epithelium is a model for the epithelial compartment of the tissue. And by using specific steps in the protocol, by giving hormones, withdrawing hormones, doing the mechanical breakdown, we can make a simplified in vitro model and protocol of the menstrual, to mimic the menstrual dynamics in vitro. And we can repeat this over and over again. We've done it more than 10 times consecutively. And we can then apply compounds and also genetically manipulate the organoids to really identify the drivers of the various processes occurring at the level of the epithelium. So for the fetal side of the story now, I'd like to tell you about an example of how we are using trophoblast organoids to study the effect of maternal signals on their behavior. And the derivation of trophoblast organoids is really, a protocol is very similar to what I showed you earlier. Essentially, we get placental tissue samples, we dissociate them to release the cells. These are embedded in this droplet and covered with a specific medium that allows these cells to grow. And over 2 to 3 weeks, we get the formation of these rather dense clusters, so they look very different to the endometrial organoids. And we can capture and allow the proliferation of the stem cells, shown here in pink, on the outside of the organoids. And inside, they spontaneously fuse to make an important cell type of the placenta, which is the syncytium, which is the site of maternal-fetal exchange and produces a lot of placental and pregnancy hormones. And in fact, if you dip a pregnancy stick into the medium, it will result positive because it's detecting the HCG that is produced by this lining. And if you go back and remember what I told you about the placenta, human placenta being very invasive, we can also generate these invasive cells in vitro by changing the culture conditions. And here you can see from outside the organoid these invasive cells crawling out. These are marked here in green by HLA-G. And these cells, as you can see here, really are highly invasive. They crawl all over the place. They actually also digest away the matrix if you leave them long enough. So in this way, we have a system that allows us to study the villus compartment for the important for maternal-fetal exchange and placental products. And on the other side, we can also study this invasive process. And in vitro, so these cells are invading the gel, but of course in vivo, they're invading through the maternal environment, and this is just an illustration to show you the villus region, and then these invasive cells more in detail as shown here, and honing in around the maternal spiral arteries. And this is an image of an in vivo section. This is again the villus compartment of the placenta, and here we see in cyan these invasive cells in the maternal environment. And it's really incredible how the extent of invasion, the number of cells, and how much they're really kind of occupying this maternal territory. And as they're doing this, they're going to be encountering many different maternal cell types, from immune cells to the surrounding fibroblasts, to the secretory cells that are lining the glands, the smooth muscle cells as they get deeper into the uterus. And so the question is, how is this maternal environment influencing this process, and how is it different in the pregnancy disorders where we see these differences in this invasive process? So I'd like to show you an example of how we are really trying to address this really in a kind of reductionist approach, again, one by one systematically, because it's a highly complex process. And this is an example of how we're trying to look at the effect of the uterine natural killer cells that are highly abundant, especially in the first trimester of pregnancy. They can directly recognize trophoblast and ask the question, what are these immune cells? What are these immune cells doing? How are they signaling to the trophoblast, and what are their effects? So this is a project that was in collaboration with Ashley Moffat at the University of Cambridge, and Roser Vento-Tormo lab at the Sanger Institute. And as part of our work, we've also generated a lot of single-cell atlases. This is data on all the different cells that are present at the maternal-fetal interface and the genes that they express that's characteristic of those cells. And we can use this data set to predict what may be those interactions happening between the maternal and fetal cells. And through computational analysis, we came up with 4 different cytokines produced by the uterine NK cells that can be detected and that the extravillus trophoblast, as invasive cells, could potentially respond to. So we just asked the question, if we provide these cytokines to these organoids undergoing this invasive process, what happens? And here is a picture again of the organoid, and you see the invasive cells coming out from surrounding the organoid. And in the presence of the cytokines, it was really clear how we had much more invasive cells. And so now we're trying to understand, what does it mean that these cytokines are basically enhancing this invasive process of the trophoblast. And so we want to, of course, to get a bigger picture of what's happening at the maternal-fetal interface. We've developed a 3D invasion assay to be able to systematically look at all the different potential signals coming from the maternal cells and their effect on the placenta. And in this setup, basically, these are tiny wells. There are 384 of them here. And you can put 1 or 2 or 3 organoids in each well, allow them to differentiate. So you see here the invasive cells coming out. And again here, these are shown here in green, surrounding the organoids. And apply individually maternal factors, and again look in an unbiased manner, what is their effect on this important invasive process? Is it inhibiting it? Is there no effect? Or is there really an enhancement of this invasive process? And this is just an example of the screen. So here you see there are many different conditions. This is a pilot screen. And then after differentiation, you see some wells where there are clearly much more enhanced invasions, some wells containing organoids that haven't differentiated at all. So we want to use this setup, which is often used for drug screening, to really understand what are the signals and how may they be different between a normal pregnancy and pregnancy disorders. So we can zoom into each one of these wells and look at a cellular level, what is the effect? So this is a control condition with no maternal signals at all. Essentially, the placental cells are inherently invasive, so if you don't give them anything, they'll just invade. And you can see the central organoid core and these surrounding invasive cells shown here in yellow. They're migrating and invading like streams, almost like a tumor. And this is an example of one of the uterine NK cytokines that I showed you, which has a really remarkable phenotypic effect. You can see the invasion is, in this case, with this specific cytokine, not as far, but even though if they invade, the cells are maintaining much more this cell-to-cell contact. And looking, for example, at a signal coming from stromal cells, which another cell type in the uterus, we see a completely different effect. So in this way, we're really hoping that we can begin to build our knowledge really from the bottom up of this highly complex process and get a better understanding of how this all works in the human context. So I showed you how we can use organoids as a model of the early human placenta. And we can study the effects of the maternal signals by applying some of these factors. And then this is amenable to a high throughput screening setting. And there are, of course, many other opportunities that these endometrial organoids and plus trophoblast organoids offer, and many other researchers in our area are really using them for asking different questions. So, for example, you can use them to study disease. So there have been organoids derived from endometriosis, endometrial cancer, and researchers have used this to look at the effect of drugs. And you can also use this to study infection. So we've heard about the dengue virus. It would be really interesting to study how that crosses the placenta. You can also use this to study and identify novel biomarkers and novel targets. And of course, as we heard this morning, of interest is really understanding these interactions with the not only between the placenta and the uterus, but also between the embryo and the uterus. And there are also opportunities for personalized medicine. And this is an area that we're really interested in because even though we do fundamental research, we're really hoping that some of the tools that we generate could one day be of translational value. And as part of this work, we've established a protocol to derive endometrial organoids from menstrual fluid. And this allows us to derive organoids in a non-invasive manner. We don't have to rely on biopsies anymore. And we've really compared from the same patient sample organoids derived from her menstrual fluid and from the biopsies, and they're really identical in the way they respond to hormones and the way they grow. So we think this is a really wonderful way to go forward because it allows us also to derive organoids from many different women from different backgrounds, and enable us to make comparisons. For example, are there hormonal differences depending on the BMI, on the ethnicity, on age, and begin to really understand how these may contribute, for example, to unexplained infertility, which may be due to the endometrial dysfunction, or for example, if there was an early pregnancy loss, is this because of some signals that were missing from the endometrial side? And going forward, we're really excited about the use of this technology. And this has really been started together with Graham Burton at the University of Cambridge and with the Bornhall Clinic, and we're really continuing it in Basel in Switzerland. So organoids are wonderful, but they're a model. They're not perfect. There's still a lot of challenges that we face to make these models better. And a lot of it also really depends on the question you're asking. So in our lab, we also like to continue to develop our model and generate different versions of it depending on the project. So for example, when we're looking at endometrial epithelium only, this is really great. It's a very reductionist system. But one of the challenges that I think we face is really trying to have an understanding of what is a normal response to hormone. We see a lot of variability across women, even women that we consider as having normal cycles. They've had a pregnancy. But so how can we establish a baseline of what's normal? And I think that requires really collecting a lot of samples across populations and studying their responses to hormones using screening platforms. And I think also as a field, we really should try to use standardized procedures that will be benefit for when we really look into translation applications. There are slightly different versions of the protocol, so I think it would be really amazing to have a dialogue on this and establish protocols that we all use. And also in this way to enable maximizing reproducibility and being able to compare the results between studies better. And I think also a major area that needs development, and this is not just in our area, is that the organoid technology is really expensive. I mean, we see this when we do these high throughput experiments. The amount of money that we spend on Matrigel is quite scary. And there are labs who are now developing synthetic hydrogels to overcome these issues, and I think this will be a very important part in also improving accessibility of these tools also to other institutes that have perhaps less access to funding. And so the bioengineering is something that we're really working on at the moment, and using this technology to improve our model systems. And particularly our interest is really studying the interactions between the maternal-fetal interface. So going back to our central question, we can use our organoids and culture them in a chip that has also those gland structures, so we can better mimic the architecture and also have access to the secretions which are trapped in the organoid spheroids. And also on the placental side, we can create that bilayer with the stem cells at the bottom and the hormone-producing layer on the top, so we can also there have better access to what it secretes. And the idea is then that we can combine these two systems and study their interactions and how they influence each other's functions in this compromise that happens during pregnancy. And this is in collaboration with Matthias Lutoff and Nicole Jagoreski labs at the IHB and Roche. So with this, I'd really like to thank my team, who have been really hardworking and really fun group of people to work with. So I'm really grateful to have them and have this opportunity also to train the next generation scientists and continue this important area of research. And I'd also really like to thank the FMI facilities, the imaging, the bioinformatics, functional genomics, and my colleagues at the FMI. They've been so welcoming and so supportive, and I'm really grateful. A warm thanks to my collaborators, and a particular thanks to Ashley and Graham, who have been wonderful mentors, and I'm so lucky to have worked with them. And I'd also like to thank the patients and the hospital staff for helping us get access to these tissues, and our past and present funders. And just a really quick advertisement, if I may, on a conference that we're organizing. So together with Amanda Clark, whom you heard her wonderful talk this morning, and Kara McKinley and Pradeep Tanwar, on the regenerative biology of the female reproductive system next year in April in 2025. So we warmly welcome you to attend. And lastly, I'd just like to finish by reiterating really the importance of understanding the fundamental biology of the uterus and the placenta, which forms the very foundation of life. And it's an exciting time to be in this field, and I hope that together we can continue to make progress and shape the world we form. And thank you.